presentation. Our next engaging session is what
Very good evening to all of you. Firstly, I would like to mention that it gives me an immense pleasure to deliver a talk from this precious platform, from where I have started my research journey as a PhD under Dr. N.C. Mishra sir in 2008. So I am thankful to the sir and the organizing committee member for inviting me for the talk. Thank you very much. Now let's start with the talk. So today's topic is production of low-cost bacterial nanocellulose, a smart multipurpose biopolymer. So as the name indicates, bacterial nanocellulose. Here the nanocellulose is of bacterial origin. It is produced from several bacterial species. This cellulose can be modified in number of ways, and it can be applied to make different bioproducts that have applicability in different techno-industrial fields. Moreover, we can recycle this nanocellulose. We can use it for the further production of the bacterial nanocellulose, as well as we can recycle the bioproducts developed from such type from the bacterial nanocellulose. So this is completely aligning with the goal of the Sustainable Development Goals to produce a greener products, sustainable products, and it is also aligning with the recently introduced BioE3 policy that relies on 3 E's: the economy, environment, and the employment, and to foster the high quality biomanufacturing. So this bio nanocellulose can be useful for variety of application. Now, if we talk in general, there are basically 2 types of nanocellulose. The first 2 types, the microfibrillated one and the nanocrystalline one, we generally obtain from the plant. It is a plant-based cellulose, okay? And this plant-based cellulose, what could be the drawback? The sources could be wood, cotton, hemp, but the production process here involves lot of chemical interference, lot of chemical processes like acid hydrolysis, bleaching, kraft pulping, etc. So the better alternative to nanocellulose could be bacterial nanocellulose. It is also known as BC, BNC, or microbial cellulose. The cellulose of microbial origin, which we can get from the bacterial species by the simple process during its metabolism that we will going to study. So bacterial nanocellulose, this BNC, it is a nanostructured exopolysaccharide secreted by a certain bacterial species. It is a pure grade cellulose. Why I am calling it pure grade? Because it is free of lignin and hemicellulose, that is the major portion in plant-based cellulose, and required to be removed by the harmful chemical process. So here we can this bacterial nanocellulose is pure grade, and there are several species responsible, bacterial organisms responsible for its production, and the model is Acetobacter. The Acetobacter is best known for the production of bacterial nanocellulose. The cellulose can be produced in the form of pellicles or ribbons or fibrils. So this is the structure. Basically it consists of lot of hydroxyl groups. So this is the organization. Here you can see this is the Acetobacter bacteria, and during the metabolism, when the media is provided, it is synthesizing the cellulose firstly in the form of these protofibrils. These protofibrils get together in a bundled form and forms the microfibrils, which ultimately turns into this hydrophilic sheet-like structure. And this sheet can be used as such for wet end applications like wound dressings, drug delivery, and it can be dried and it can be used for the packaging application. It has potential for the use for different techno-industrial application. So this is the detailed process of biosynthesis of this bacterial nanocellulose, which is an extracellular polysaccharide from the bacteria. So when the bacteria metabolize the glucose, it produces glucose 6-phosphate, finally glucose 1-phosphate, and finally the uridine diphosphoglucose, which is the ultimate precursor for the cellulose synthesis, bacterial nanocellulose synthesis. And this is how the subunit get clustered by the strong hydrogen bonding, and we can get a pellicle. Now, why this bacterial nanocellulose is important, or why we use it, what is alike than its plant-based counterpart. So this BNC have some implausible property that makes it useful for different agro-industrial applications like high absorbency and resistance to fiber lift. So its water retention value is quite high. It can absorb and retain water, and this application is useful in patent application like drug delivery and wound dressing. Wet strength is high. It is inert, so it can be used for the. Sir, I'm going to the. Yeah. So, and then it can withstand high temperature. It is biocompatible, biodegradable, and generally recognized as safe. So these are different application areas like cosmetics, biomaterials, food industry. These are certain uses of the bacterial nanocellulose as coronary vessel implants or as a wound dressings. So, but if it is a polymer of such importance, what are the challenges? What is the research gap? The research gap is that its production is quite costly and its yield is quite low. Okay, so it can. What could be the possible solution? The possible solution is the use of alternate substrate or alternate carbon source, and the use of different alternative strains other than the commercially available ones. So basically my work is focused on the use of the cost-effective substrate and alternate organelles for the production of BNC. So in literature there are a lot of work which have used the carbon sources like Okay. So my objective is the production of bacterial nanocellulose by the static intermittent fat batch and bioreactor based approach using black tea broth as a cost effective media, and the symbiotic combination of bacteria and yeast as a co-culture. Then its characterization and finally its application potential towards packaging and biomedical application. So here why I have used that black tea broth, because black tea broth is a cocktail of different organic acids, vitamins, minerals, and what brings fermentation in this brewed tea, and what is the I have used the symbiotic combination of bacteria, because here the yeast component is present that will ferment the ethanol, and this ethanol is in turn used by the acetic acid bacteria, and finally it will synthesize the cellulose. These are the common species that synthesize cellulose that is present in the black tea. So the methodology adopted is the static batch production and static intermittent fat batch production. So we have got the good yield when we have switched the process from static batch to static intermittent fat batch. And here we have also produced bacterial nanocellulose using the Scooby culture and black tea media in rotary disc bioreactor. So this is a bioreactor, the fabricated bioreactor, and here you can see the nanocellulose synthesized over it. So this is the gist of overall methodology. Then we have evaluated the dry weight, the yield and production kinetics. So here you can see in the static production, the yield is only 13% around, it is 13 gram per liter. However, when the process is switched to the static intermittent, the yield has increases to around 29 gram per liter. And this is the production kinetics. And here this is the production using the RDBR reactor. Here we have got the negative result because the Scooby culture has not supported the cellulose synthesis in RDBR, maybe due to the rotation of the disc. So these are the water retention results of the BNC formed under different conditions. So it is around 90. It is highest in RDBR, and it ranges from 83 to 93%. This is the SAM image. Here you can see the protofibers coming out of the cell. And here these are other physicochemical characterization, the FTIR, XRD, and here you can see the TGA analysis. This shows that the BNC is quite stable up to around 300 degrees C, and mostly it is crystalline in nature. Its crystallinity is around ranges from 68 to 79 in different condition. So this is the overall result. When we have used the black tea broth under the static fermentation, the yield is 13.6, which increases to 29.4% in under static intermittent fat batch. However, there is almost negligible yield in RDBR. So, and this is the substrate to BNC conversion rate. The residual sugar, though it is highly used in case of RDBR, but the product is not formed. Okay, so maybe it can get converted to some other form. So the best one is the static intermittent fat batch technique for the production of BNC. So this study is published in International Journal of Biological Macromolecule, and now there are we have modified this bacterial nanocellulose, the cost-effective bacterial nanocellulose by chitosan, and here you can see the inhibition zone in 1 gram positive and gram negative bacteria, and we have utilized it for the packaging application. So here you can see the result on 20th day. There is no wrinkling when we have used the BNC plus chitosan plant film. However, in unwrapped tomatoes, it shows wrinkling on day 9 and then microbial spoilage. So here we have also modified it by using different bioextracts, and we have seen the result. The best result we have got for the tea Arjuna, the best antimicrobial susceptibility we have got for this. And here we have utilized this the composite of bacterial cellulose, chitosan and gelatin for tissue engineering application, and you can see the cells growing, the proliferating over the scaffold. So the conclusion is the static intermittent fat batch strategy along with FTTB fermented black tea broth medium could be used as a low-cost medium for the BNC production. Mixed culture could be more beneficial in comparison to the single commercial strain like Acetobacter. RDBR could be useful for large-scale production, but it is not working in case of BTB. And antimicrobial bacterial nanocellulose films using bioextracts have the potential to be used as wound dressing or antibacterial textile for hospital environment. Next, this bacterial nanocellulose chitosan and its composite freeze-dried film show potential in vitro L9 to 9 fibroblast response and have potential to be used for tissue engineering application. And finally, BNC production and its bioproducts usage aligns and could meet with the UN Sustainable Development Goal and Bio-E3 policy. So I like to acknowledge Department of Science and Technology, where I have first started my study on BNC, and Avantha Center for Industrial Research and Development, and finally I'm moving towards further towards the application part, and I am working for this at University Center for Research and Development, Chandigarh University. These are some latest publications. These are the list of my collaborators. This is my small research team.